For decades, the fight against Alzheimer's disease has been dominated by a single story, the story of a protein called amyloid beta. The idea, known as the amyloid hypothesis, was simple and compelling. This sticky protein clumps together in the brain, forming plaques that kill neurons and cause dementia. For years, the strategy was clear: get rid of the amyloid, and you cure the disease. But after countless clinical trials and billions of dollars, the results have been, to put it mildly, complicated. We now have the first generation of drugs that can clear amyloid plaques from the brain, a monumental scientific achievement. Yet they aren't a cure. They slow cognitive decline, but only modestly. This has forced a painful but necessary reckoning in the scientific community. The verdict is in: amyloid is guilty, but it didn't act alone. Today, we're going to explore the future of Alzheimer's treatment, a future that looks beyond amyloid to hunt down its many co-conspirators. The story is no longer about a single villain, but a complex criminal network, and understanding it is the key to finally defeating this devastating disease. The next major frontier in Alzheimer's therapy is targeting another protein, one you may have heard of: tau. Inside a healthy neuron, tau proteins act like the ties on a railroad track, stabilizing the cell's internal transport system, the microtubules. These tracks are essential for shipping nutrients, neurotransmitters, and other vital components from one end of the neuron to the other. But in Alzheimer's, something goes wrong. The tau proteins become chemically altered, they detach from the tracks, and then they clump together inside the neuron, forming what are known as neurofibrillary tangles. These tangles are devastating. They cause the internal railroad to collapse, disrupting the neuron's ability to function and communicate. Eventually, the neuron dies. What's particularly insidious is how tau pathology spreads. It follows a predictable pattern through the brain, almost like a contagion hopping from one neuron to the next. In fact, the progression of tau tangles through different brain regions, known as break staging, correlates much more closely with the severity of cognitive symptoms than the burden of amyloid plaques does. This suggests that while amyloid might start the fire, it's the spread of toxic tau that really burns the house down. The logical next step, then, is to develop therapies that specifically target tau. Researchers are pursuing several strategies. Some drugs are designed to prevent tau from clumping together in the first place. Others are antibodies that aim to capture and clear the toxic tau proteins as they travel between cells, stopping the spread. Still others aim to stabilize the microtubules that tau has abandoned. Combining an amyloid-clearing drug with a tau-targeting drug could be a powerful one-two punch, tackling both the trigger and the primary agent of destruction. But the conspiracy runs even deeper. The brain doesn't just sit there passively while amyloid plaques and tau tangles accumulate. It fights back. The brain has its own dedicated immune system, and the primary soldiers in this system are cells called microglia. For a long time, the inflammation we see in the Alzheimer's brain, or neuroinflammation, was thought to be just a consequence of the damage caused by plaques and tangles. But we now understand that it's a critical driver of the disease itself. Microglia are the brain's housekeepers and security guards. In a healthy brain, they are constantly patrolling, clearing away cellular debris, and gobbling up potential pathogens. Early in Alzheimer's, they try to do their job, attempting to clear the accumulating amyloid proteins. But they are overwhelmed. The constant, chronic stimulation by amyloid plaques switches them from helpful housekeepers into a state of angry, chronic activation. Instead of cleaning up, these activated microglia start releasing a flood of inflammatory molecules, like cytokines. This sustained inflammatory response creates a toxic environment that directly damages neurons, exacerbates tau pathology, and can even weaken the blood-brain barrier, a critical protective shield that separates the brain from the rest of the body. In essence, the brain's own defense system turns against it, contributing to the very neurodegeneration it's supposed to prevent. This has opened up an entirely new therapeutic avenue, modulating the brain's immune response. The goal isn't to switch off inflammation entirely, because we need that initial cleanup response. The goal is to finely tune it, to coax the microglia back into their protective housekeeping state, while calming their destructive inflammatory rage. A key player in this process is a receptor on the surface of microglia called TREM2. TREM2 helps microglia recognize and clear harmful substances, including amyloid. Mutations in the TREM2 gene significantly increase the risk of developing Alzheimer's, highlighting its crucial role. Therefore, drugs are now in active investigation that can boost TREM2 activity. The idea is to create supermicroglia, cells that can efficiently clear pathological proteins without causing the collateral damage of chronic inflammation. This brings us to one of the most important paradigm shifts in how we think about treating Alzheimer's: the move towards combination therapy. The era of searching for a single silver bullet is likely over. Alzheimer's isn't one problem. It's a cascade of multiple interconnected failures. Amyloid builds up, which triggers tau pathology, which in turn fuels chronic neuroinflammation. At the same time, the brain's vascular system might be failing, and its cellular waste disposal systems might be clogged. Attacking just one of these problems is like trying to fix a sinking ship by plugging only one hole. The future of treatment will almost certainly involve a therapeutic cocktail, much like the combination therapies that turned HIV from a death sentence into a manageable chronic condition. Imagine a patient receiving a treatment that includes one drug to clear amyloid, a second to stop the spread of tau, and a third to modulate the microglial response and reduce inflammation. For a patient with significant cardiovascular risk factors, you might add a fourth drug to improve blood flow and strengthen the blood-brain barrier. This multi-pronged attack on the disease's core pathways holds far more promise than any single target therapy ever could. It acknowledges the true complexity of the disease and attacks it from all sides. But even the most effective drug cocktail has a critical limitation. It works best when the damage isn't already done. The clinical data from the new amyloid-targeting drugs have made one thing crystal clear. Interventions are most effective when started very, very early in the disease process, long before widespread, irreversible neurodegeneration has occurred. By the time a person shows symptoms of memory loss, their brain has already been under attack for perhaps 10 to 20 years. The plaques, the tangles, the inflammation, they've had a massive head start. This is why the single biggest revolution in the fight against Alzheimer's may not be a drug at all, but a test. We are on the cusp of a new era of diagnostics. For years, the only way to definitively diagnose Alzheimer's was through a brain autopsy after death, or through expensive and invasive procedures like PET scans and spinal taps. This is changing, and it's changing fast. Scientists have developed highly sensitive biomarkers, especially blood tests, that can detect the telltale signs of Alzheimer's pathology, amyloid, and specific forms of tau with incredible accuracy. Imagine a future where, as part of your routine physical at age 50, a simple blood test can tell you if you have the earliest signs of Alzheimer's pathology building up in your brain, years or even decades before you would ever notice a symptom. This would be a complete game changer. It would transform Alzheimer's from a disease we diagnose at its end stage into a condition we can identify at its very beginning. It would allow us to intervene when the brain is still resilient and therapies are most likely to succeed. It shifts the entire focus from treatment to prevention. Identifying at-risk individuals decades in advance opens the door to truly preemptive strikes, potentially stopping the disease cascade before it ever leads to cognitive decline. This leads directly to the final piece of the puzzle: personalized medicine. As we've seen, Alzheimer's is not a monolithic entity. The combination and relative importance of its drivers can differ from person to person. One individual's disease might be primarily driven by a strong genetic predisposition for amyloid accumulation and inflammation. Another's might be more heavily influenced by vascular problems, like high blood pressure and cholesterol, which lead to a leaky blood-brain barrier and reduced clearance of toxins. It makes sense that these two individuals might benefit from different therapeutic combinations. As our diagnostic tools become more sophisticated, we won't just be able to say, "You have Alzheimer's." We will be able to say, "Your Alzheimer's is characterized by high amyloid, moderate tau pathology, and significant vascular dysfunction." This detailed biological fingerprint will allow for the tailoring of treatments. The patient with the strong inflammatory component might receive a potent microglia modulator, while the patient with vascular issues might get therapies focused on healing the brain's blood vessels. This personalized approach moves away from a one-size-fits-all strategy and towards precision medicine, maximizing effectiveness and minimizing side effects for each individual. So, where does this leave the amyloid hypothesis, the theory that started it all? It would be easy to dismiss it as a failure, a costly distraction, but that would be a profound misreading of the history of science. The amyloid hypothesis, for all its controversies and setbacks, has been invaluable. It provided a concrete, testable framework that focused research and investment for decades. It was a hypothesis that had to be tested. Its trial in the crucible of clinical science was painful and expensive, but it has forced the field to confront the true, staggering complexity of Alzheimer's disease. We now know that amyloid is a key initiator, the first domino to fall. But we also know that focusing solely on that first domino is not enough. We have to stop the entire chain reaction. The future of Alzheimer's is a comprehensive strategy. It's about using combination therapies to fight a multi-front war against amyloid, tau, and neuroinflammation. It's about deploying revolutionary blood tests for early diagnosis, shifting the battleground from late-stage treatment to early-stage prevention. And it's about using personalized medicine to tailor those interventions to the unique biology of each patient. Amyloid was not the full story, but it was the essential first chapter. By proving that amyloid was a guilty party but not the only one, we have finally been forced to identify and bring to trial its many co-conspirators. The path forward is clearer now than it has ever been, moving us closer to a future where dementia is no longer an inevitable consequence of this disease, but a preventable and manageable condition.